Bismillahirrahmanirrahim. We start another paper from the June 23 series, but this is now the O-level biology 5090, and this is the question paper 1-1, which is one of the variants, and we're going to discuss the questions of this paper. As you all know that this paper is for 1 hour, and this is the June 2023 paper, and now this has remained. This is of course the first paper that we are doing for the new syllabus, because the new syllabus started in June 2023. So this is the first paper that we're going to be talking about. Now, if you look at the first question, it says which description applies to a red blood cell. Now, you know red blood cell is a human cell, so it would not have a cell wall. So there will be no cell wall. So the only two answers which are correct. Now, only in the red blood cell there is no nucleus, but in otherwise every other animal cell does have a nucleus. So that is why the answer is A. A green leaf plant produces spores. To which group does the plant belong? Produces spores. Now this is of course a new syllabus, and that is why the answer to this is C, because ferns produce spores, and that is part of the classification question which has now been added to the new syllabus. So this would have been a little challenging for the students, because if they don't remember that ferns produce spores, you see dicotyledons do not produce spores, they have seeds. Fungi also have some sort of spore-like structures, but they are not actually this, just spread by other mechanisms. So monocotyledons again are seeds. Now coming to question number 3, which statement is always correct for both diffusion and osmosis? You know the definition of osmosis is the movement of water molecules. Diffusion can be anything. Oxygen can diffuse, carbon dioxide can diffuse, glucose can diffuse. So which statement is always correct for both diffusion and osmosis? They depend on energy from cells. You know, these are passive processes. They do not rely on energy. They involve the movement of particles down a concentration gradient. Yes, diffusion is down the concentration gradient, and osmosis is down the water potential gradient, but that is a sort of a concentration gradient. That is why the answer is B. They involve the movement of water molecules. Now this is not correct, because osmosis only involves the movement of water molecules, not diffusion. Diffusion can be anything. So that is why C is incorrect. They take place only in liquids. No, gases can also diffuse. Gases can also diffuse. So it says which is correct for both diffusion and osmosis. Now coming to question number 4, this is a little difficult for some students who do not understand graphs very well, and I was just explaining this today to one of the students on WhatsApp. The graph shows the effect of oxygen on the uptake of ions by two sets of barley plants. Uptake of ions by barley plants, I always say put some figures here. Like I put here 1, 2, 3, 4, 5. So the uptake of ions is increasing. Now in this situation, the graph goes like this. Low concentration of oxygen available, but the graph for high concentration of oxygen is goes up more. So uptake of ions is more. So if he says 5 uptake of ions, in this situation the uptake of ions is only something like 2.5 or something like this. So always explain the graph to yourself. This is a very important way of doing graph questions. Some of you really struggle with this. Except for oxygen availability, both sets of barley plants were grown in identical conditions. Which process is taking place to explain the difference between the two curves? You see, why would more oxygen result in a higher concentration of uptake of ions by the barley plant? Because oxygen is used only in respiration, aerobic respiration, and in aerobic respiration releases energy, and energy is needed neither for diffusion, neither for osmosis. Photosynthesis is not in the question because they are not talking about that, is uptake of ions. So the answer is active transport. Active transport is against the concentration gradient, and it is requires energy, and energy is produced by either aerobic respiration or anaerobic respiration. But here, because oxygen availability is we're talking about, so that means it is aerobic respiration. So the answer to 4 was A, active transport. What process is taking place? What process is taking place to explain the difference between the two curves? More oxygen means more respiration, more energy release, more active transport. Then coming to question number 5, a student tested a sample of food. The results are shown. Color after adding Benedict solution and heating was red. Color after adding purple was protein is present. So P for purple, P for protein, and P for present. Red means this is a reducing sugar. And color after adding iodine remained brown. So that means there is no starch. Because if it remained brown, it only turns blue-black. If iodine turns blue-black, then starch is present. So now it says which nutrients were in the food. Now, of course, it has to be protein and reducing sugar only, because there's no starch. Here it said protein, starch, and reducing sugar. How is there starch? The iodine was yellow-brown, so no starch. So wherever starch was the answer, that was wrong. You could have easily figured that out as well. Wherever there was starch, that was the wrong answer. Only the one without starch was the correct answer. Question number 6. The graph shows the effect of substrate concentration on the rate of an enzyme controlled reaction. The results have been obtained using a fixed concentration of enzyme at a temperature of 30 degrees Celsius. Okay, now I'm going to draw a diagram for you to understand this. Now you can see I've drawn a diagram for you to see this. Which statement explains the shape of the graph from X to Y? Now in this situation, let's look at this situation first. At this situation now, these are probably 10. So 10 substrate molecule forms, 10 product formed. And then we keep on increasing this, 20. And then we keep on increasing this, 30. So if you keep on increasing this and a point comes when this is also 100, that is when it's going to level off. Because if you increase this to 120, now the enzyme is the limiting factor. Previously when this was 10, then enzyme, the substrate was the limiting factor. But now when you have it 100, that means all the active sites are occupied. And if you have it increased now, the rate will become constant. The rate of reaction will become constant. The rate of reaction will become constant. But if you increase it to 1000, now they'll all line up the substrate. So 100 fit, 100 product formed, 100 fit, 100 product formed. Now it says the results have been obtained using a fixed concentration of enzyme at a temperature of 30 degrees Celsius. Which statement explains the shape of the graph from X to Y? Denaturation of the enzyme molecules is increasing. The temperature is 30. Temperature has not changed. The enzyme active sites are not all occupied. You realize here at this point and at this point that the graph is still increasing. So they are probably at this point they are less than 100. First they were 10, then they were 20, then it was 40. So it is still here in this situation, it's still not 100, because then it will level off if it becomes 100, because the rate of reaction will become constant, right? So that is why the answer is B. The enzyme active sites are not all occupied. The concentration of the substrate molecule is decreasing. No, the substrate concentration is increasing. You can see it here. This is the line which I have given you. The frequency of effective collisions between enzyme active site and substrate molecules is decreasing. No, the substrate is increasing. So the frequency of effective collisions is increasing because the substrate is increasing. Here you had the substrate was say 5, then 10, then 15, then 20, then 25, and a point will come and it will be 100 if there are 100 enzymes or 100 active sites. So please understand this question. This is a very, very important question of how we're going to interpret graphs and how we're going to understand graphs. Question number 7. The graph shows how the rate of photosynthesis in a plant varies with light intensity. So the light intensity is increasing like this. So here say we have got a 100 watt bulb, then 2, then 3, then 4, then 5, so on and so forth. And the rate of photosynthesis is say 5, then 10, then 15, then 20, and so on. So what you have to understand is that they are going to ask you a question on this area QR. So when you increase the light, the rate of photosynthesis is increasing a bit. Now it's increasing a little more. Now it's increasing a little more. And after this, if you increase this, the rate of photosynthesis is constant. If you press the accelerator of a Mehran, it will only go at a certain speed. If you press the accelerator fully of a Ferrari, it will go at a very, very high speed. So now the rate has become constant. Which statements are correct? Light intensity is the only limiting factor in photosynthesis. No, the other factors, temperature, carbon dioxide concentration. Between points P and Q on the graph, light intensity is a factor that is limiting the rate of photosynthesis. Between Q and R, P and Q, sorry. Between P and Q, so between this area, so this point when this is increasing, yes, light intensity is the limiting factor because here light was less, so less rate, then increase, more rate, then more, then more. So it is slowly increasing as the light intensity is increasing. So 2 is correct. Between Q and R, another factor is limiting. Yes, now the light intensity is not increasing, so the rate has become constant. Now some other factor is limiting. Could be temperature, could be CO2 concentration. The factors that affect the rate of photosynthesis are TLC: temperature, light intensity, and CO2 concentration. So what you have to understand is that these are the three factors which emit the rate of photosynthesis. So that is why the answer to this is C, which is 2 and 3, which are correct. Between P and Q, light intensity is limiting. Between Q and R, there another factor is limiting the rate of photosynthesis. Now question number 8. The diagram shows part of a transverse section through a leaf. So we have 1, which is the xylem and the phloem, which is the vascular bundle. 2 is the spongy mesophyll cells or the spongy mesophyll area tissue. And then 3 is the palisade mesophyll cells, which of course contain a large number of chloroplasts. The spongy mesophyll contains less chloroplast, and of course in the vascular bundle there are no chloroplasts. So what are the main functions of the three labeled regions? So one is photosynthesis, no, one is transport. Why? Because this is the vascular bundle. So it's got to be transport. So it's either B or C. And then it says region 2 is what? Region 2 is the spongy mesophyll. So that is gas exchange. So that is why the answer is C. Because region 1 is transport, region 2 is gas exchange, and region 3 is photosynthesis, because palisade mesophyll cells have a lot of chloroplasts. And so the major function of that area is photosynthesis. Chloroplast means chlorophyll, chlorophyll means light, and chlorophyll is going to result in CO2 plus water gives you glucose plus oxygen. That's photosynthesis. Now question number 9, which description of the function of root hairs is correct? I find this a very weird MCQ because I mean, how could a student get it wrong? Is that osmosis is only and only of water? Osmosis is only and only of water. So root hairs absorb ions by active transport and osmosis. How can ions be absorbed by osmosis? So this is definitely wrong. Root hairs absorb ions by osmosis only, again wrong. Ions is by active transport. Root hairs absorb water by active transport. That's absolute rubbish. How can water be absorbed by active transport? So now we are left with D, which is the correct answer. Root hairs absorb water by osmosis. Then which conditions result in the highest rate of transpiration from a plant? So you have to remember is that this is a plant, right? Why would water leave the plant? Why would water vapors exit the plant? The humidity has to be less and the temperature has to be high. The humidity has to be less. Why? Because water, in order to diffuse out, must be more inside and less outside. So water vapors inside the leaf should be more than the water vapors outside. So humidity should be less. Now the humidity means it should be 60 and the temperature should be high. So that is why the answer is B, that 60 humidity and 25 is the higher temperature, because higher the temperature, more the rate of evaporation, more the water vapor being formed, and the higher the rate of diffusion. Temperature increases kinetic energy, increases the rate of diffusion. So that is why question 10, the answer is B. Question 11, which disease can be the result of dietary deficiency of both a mineral ion and a vitamin? So mineral ion is calcium, that's the syllabus, vitamin D, and the answer is rickets. The deficiency of calcium and vitamin D results in rickets. Okay, anemia is because of the deficiency of iron. Diabetes is the deficiency of making insulin, so won't be making any insulin, so the person will suffer from diabetes. And scurvy is vitamin C, deficiency of vitamin C. A student states the contraction of circular muscle pushes the contents forward. Which process is being described? In circular muscles, contracting is only in peristalsis. So the answer is A. Deamination takes place in the liver. That's removal of the nitrogen part. Translocation is the movement of the sucrose in the phloem, and transpiration is the movement of diffusion of water vapors out of the leaf. So that is nothing to do with muscle contraction. Deamination is the only thing which is human. Translocation and transpiration is in plants. Contraction of circular muscle is something which is to do with humans. Plants do not have muscles in them. Then coming to question number 13. An investigation was carried out on digestion and absorption in the alimentary canal. The diagram shows the apparatus used. The whisking tubing is permeable to small molecules such as glucose, but not to large molecules such as starch. So glass rod to support the whisking tubing beaker, water at 36 degrees Celsius, starch solution and amylase. Now you have to understand is that starch will be broken down by amylase into maltose, and maltose is going to diffuse out. So maltose is going to diffuse out here. Starch cannot diffuse out because it is said to you that the whisk is permeable to small molecules, but not to large molecules. So starch is going to stay in here. So let's color red the starch. So the starch stays in here. Starch cannot come out. But then the starch is also going to be broken down to maltose. But the maltose can come out. Maltose can be present in this water at 36 degrees Celsius. So which color? After 1 hour, samples of water in the beaker. So samples of this water is taken out and tested with Benedict's and with iodine solution. Now, what are the colors which will be obtained? Naturally, the reducing sugar test will be positive, so that has to be red. And the iodine color will remain yellow-brown because there's no starch in it, so it cannot turn blue-black. So that is why the answer is D then, because starch cannot come out. Starch has been hydrolyzed by the amylase and maltose has been formed. Maltose has diffused out. So maltose is in this water. We've taken out this water. We've taken this water out and we have analyzed it and we have heated it with Benedict solution, and we have got either a blue, if they've given you these options, blue, red color. So the blue means no reducing sugar in it. Red means reducing sugar present. If you don't understand this, you need to revise your food tests. Question number 14. Some students were given this apparatus shown. There's the mouthpiece, there's a rubber bung, there's a glass tubing, there's lime water in both, there's test tube 2 and test tube T and test tube U. Now what you have to understand is how this air is going to come in. Now the air comes in through this and then dips in here and then goes into your mouth, and this is when you inhale. When you exhale, I'm just using another color for the exhale, then this part goes in and is going to go in and dip into the lime water. And this lime water will turn milky. Why? Because it is the exhaled air. This is the inhaled air. So this lime water remains clear. This one remains clear. This one will turn milky or become cloudy or milky, whatever you want to say. So then this exhaled air is going to be going in here and then is going to go out of this here. This is the way you've got to understand. Inhaled air coming in. This is, I've explained this to you in my videos on this chapter. They place the mouthpieces in their mouth and they gently breathe in and out through the apparatus 10 times. Which statement describes what happened when they breathed in and out? Which statement describes what happened when they breathed in and out? When they breathed in, air from the room bubbled through test tube U. No, that's wrong. When they breathed out, air from their lungs bubbled through test tube T. T again, this is wrong. When they breathed in, air from the room bubbled through test tube T and the lime water remained clear in test tube T. So, you see, this is what you have got to understand. If you don't understand this, you can't do this question. So the correct answer is C. When they breathed in, air from the room bubbled into T and the lime water remained clear. Why? Because inhaled air has less carbon dioxide. Inhaled air has less CO2. But exhaled air has more CO2. Exhaled air has more CO2, which is on this side, which is on test tube U. And the last possible answer was when they breathed out air from their lungs, bubbled through test tube tube. When they breathed out air through test tube and the lime water remained clear. No, the lime water here becomes cloudy because exhaled air contains more carbon dioxide. So please understand this first. And unless you understand this, how air is going to be inhaled and exhaled, you cannot really do this question. How many molecules of carbon dioxide will you produce from the breakdown of two molecules of glucose in aerobic respiration? Okay, so let's do the equation. So question 15, the equation is C6H12O6 plus 6O2 gives you 6CO2. So how many two molecules of glucose? So if one molecule of glucose gives you 6 CO2, so then there will be 12 CO2 and two molecules of glucose. That's why the answer is D. Now coming to the question number 16. Which equation shows the type of respiration that can cause an oxygen debt in muscles? Now the answer has to be lactic acid, because the type of respiration that causes anaerobic respiration in the muscles when you do strenuous exercise, that's when lactic acid is produced. This is an equation for aerobic respiration. This is an equation for anaerobic respiration in yeast, where ethanol and carbon dioxide are produced. This is of course nowhere this happens, so this is all just rubbish. But this is just to distract you, so that if somebody doesn't know this, is just going to do it by mistake, is saying, oh yes, lactic acid and carbon dioxide, because if you don't know your biology very well, that is the problem. Now before I start the next question, I want you to look at these graphs. Now this is the most important graph which I want you to see, is the pressure graph. You see, this is the pressure which is in the aorta, then pressure in the arteries, then you can see the pressure in the capillaries. Now this is the area which has been defined. This is the arterioles and this is the capillaries, and then you can see the veins and then the vena cava. So the pressure is decreasing. In the capillaries it has decreased a lot, but in the veins it has decreased even more than in the veins and the vena cava. Now the velocity is slightly different. I want you to know the velocity thing also. The velocity is the same, then it decreases as it goes in the arterioles, and then in the capillaries the velocity is very very low, 0, and then the velocity slightly increases in the veins and the venules. But if you look at the cross-sectional area, the area is very interesting. The area is uh, very little here, but then it has greatly increased in the capillaries, and then it is again greatly decreased in the veins. So the cross-sectional area is the most in the capillaries. This is the capillary graph. I want you to pause the video here and have a look at this graph before we do the question. Another graph showing you these pressure and the vessels. I want you to see this graph also. I want you to see what is on the y-axis, blood pressure in millimeters of mercury, and then you can see the arteries, arterioles, and the venules and the veins. So pause the video here, have a look at this, understand this. Question number 17, blood pressure was measured in the pulmonary artery, pulmonary capillaries, and pulmonary vein. Which blood pressure results could be correct? So 1485. The answer is A. The reason being is that blood pressure in the pulmonary artery is the most, and in the capillaries is less than that, and the vein it's much lesser than that. The other ones are all incorrect. Which component of blood engulfs pathogens? Which component? So that is phagocytes, phagocytosis. Phagocytes engulf, digest, and destroy pathogens. Then question number 19, what would help reduce transmission of malaria? Malaria is caused by a protozoa. It is spread, the vector is the Anopheles mosquito, the female Anopheles mosquito. So 19, use of a condom would prevent sexually transmitted diseases. Use of antibiotics would be given as a treatment for bacterial diseases. Mosquito nets, yes, that's it. Washing hands would be more in the cholera, towards the cholera side. So you're washing hands. So the answer was 19, the answer was C. Then coming to question number 20, the table shows the death from lung cancer in the country among smokers and non-smokers. Average number of cigarettes smoked per day, 0. Death from lung cancer per year per 100,000 people, 10. Then number of cigarettes increased 1 to 14. Then 78 people die of cancer per year. Then 15 to 25. Then 127 people die per year out of 100,000 people, and then if you smoke 26 or more, then 251 people die. What can be concluded from the data? Now, when you're doing this question, what you have to do is you have to read in a different manner. You have to think, you have to ask your question. Does the data tell me that people who get lung cancer are likely to be smokers of 26 or more cigarettes per day? Does the data tell me people who do not smoke will not get lung cancer? See, people who smoke have a higher chance of getting cancer. This looks a little correct. Smoking is the only cause of lung cancer. No, the data doesn't tell you anything about anything else. It just tells you the average number of cigarettes smoked per day, and it tells you the deaths from lung cancer per year per 100,000 people. So it's not telling you anything else. Now, don't think that it's telling you something which is not telling you. What can be concluded from the data? The conclusion is only that people who smoke have a higher chance of getting lung cancer. We have not investigated any other things. Was it because of pollution? Was it because of something else? Maybe they ate a lot of burgers, or maybe they ate a lot of chicken kadai or something like that. No, nothing's been mentioned in the data about anything else. So there is no question of people who do not smoke will not get lung cancer. There is zero smokers also have death from lung cancer. That is also something which is there in the data. So that doesn't tell you people who do not smoke will not get lung cancer. So always ask yourself this question: does the data tell me? Then read A. Then does the data tell me? Read B. So this is one way of doing this question. Then coming on to question number 21. What is one way the development of antibiotic resistant bacteria can be minimized? Now this is a very important syllabus point. Use of antibiotics only when essential. Use of antibiotics only when essential. Sharing of antibiotics with family members, no, that's going to increase it, because the family member might not need an antibiotic, and you just say, okay, well, I was taking this medicine, you also take it. Then use of antibiotics to prevent infection. The infection could be a viral infection, could be a bacterial infection. That is wrong. You don't use antibiotics in viral infections. You do not share it with family members. Use of antibiotics to treat all infections. No, there are viral, fungal, bacterial, protozoal infections. Malaria is called by a protozoa. You don't take antibiotics for that. So that is why A, C, and D are wrong. Please understand why it is wrong. Don't just say it's wrong because you just because I'm saying it's this is, but you must understand why it's wrong. The last question of this video, question number 22. What are the effects of the human deficiency virus on the immune system? Immune system becomes weaker, antibody production decreases, and number of lymphocytes decreases. Yes, that's the answer. Because you have to understand is that HIV, of course, is going to attack the helper lymphocytes, and the helper T lymphocytes, of course, will result in less antibody production because it also affects the B lymphocytes. So less antibodies produced, less number of lymphocytes because it destroys the lymphocytes. The virus goes and invades the lymphocytes. So the answer is A.